Hi all, welcome back to our Allied Lecdem channel. In this video, we are going to see about neuromuscular junction. What is neuromuscular junction and its structure and neuromuscular transmission and so on. So let's get into the video. First, we'll see what is neuromuscular junction. Neuromuscular junction is a synaptic connection between the terminal end of a motor nerve and muscle. In simple, neuromuscular junction is a junction between terminal branch of the nerve fiber and muscle fiber. And it is also the site for the transmission of action potential from nerve to the muscle fiber, and it is also site for many diseases and site of action for many pharmacological drugs. Before entering into the topic, we should know about what is motor neuron and motor unit. Motor neuron is nothing but the nerve that innervates the muscle fiber, that is a nerve which supply to an organ. In this context, we are speaking about muscle fiber. Then what is motor unit? Motor unit is nothing but a single motor neuron and muscle fiber it innervates. So it combines both motor neuron and muscle fiber. It has been stated as motor unit. We'll see about anatomy of neuromuscular junction, that is the structure of the neuromuscular junction. So skeletal muscle fibers are innervated by a motor nerve fiber, that is each nerve fiber, that is we are speaking about axon, which divides into many terminal branches. So each terminal branches innervates, that is each terminal branch supply to one muscle fiber through a neuromuscular junction. Okay. First structure we are going to see is axon terminal, which is also known as motor end plate. The terminal branch of nerve fiber is called axon terminal. So, when the axon comes closer to the muscle fiber, it loses the myelin sheath. Myelin sheath is nothing but the covering which covers the axon. So, the axon cylinder, which is exposed. So this portion of axis cylinder is expanded like a bulb, which is called motor end plate. And this motor end plate has two specialization. The first specialization is that it has a junctional fold, that is invagination, a groove which is being present in the sarcolemma that provide a large surface area where acetylcholine from the synaptic end bulb can interact. So it has a groove-like structure in the presynaptic membrane, that is motor end plate is also known as presynaptic membrane. So this invagination inside this muscle fiber is known as synaptic trough or gutter. Synaptic trough or gutter. The second specialization of the axon terminal is this folded like structure in the sarcolemma contains 30 to 40 million acetylcholine receptors. These receptors are integral transmembrane protein that function as an ion channel 1 activator. That is, when there is a change in the action potential, once the ion channels are activated, this acetylcholine receptors are being activated and it helps in release of acetylcholine. Now a question rises, from where these acetylcholines are being synthesized? So the axon terminal, that is motor end plate, as mitochondria. So when this mitochondria contain ATP, which is a source of energy for synthesis of acetylcholine. So acetylcholines are being synthesized in the mitochondria. As we could see in this first image, the presynaptic terminal is being marked as a green in color, which is the upper part. So a postsynaptic terminal in the second image, we could see it has been marked as a green in color at the lower side. So the third image is in neuroreceptor, the synaptical vesicle, that is contain acetylcholine, that is purple round like structure. As we could see in this image, it's a purple round like structure. First it enters the synaptic trough, okay, and reaches the receptor present in the postsynaptic membrane. So the acetylcholine from the synaptic vesicle, it reaches the synaptic cleft. We'll see what is synaptic cleft, and it reaches the neuroreceptor present in the postsynaptic membrane. Next is synaptic cleft. We saw the membrane of the nerve ending is called presynaptic membrane. The membrane of the muscle fiber is called postsynaptic membrane. So the space between these two membranes, that is presynaptic and postsynaptic membrane, is known as synaptic cleft. So synaptic cleft contain an acetylcholinease, an enzyme known as acetylcholinesterase. So, which helps in breaking down of acetylcholine into acetyl and choline, neither of which can activate the acetylcholine receptor. So, as we could see in the image, the space between presynaptic and postsynaptic membrane, which has been marked in green color, is a synaptic cleft. Next, we'll see subneural cleft. As we saw in presynaptic membrane, our invagination structure, which has been known as synaptic trough or gutter, same as that of that, if the invaginations or numerous folds which has been thrown into a postsynaptic membrane is known as subneural cleft. Okay, subneural cleft and then postsynaptic membrane contain a receptor called nicotinic acetylcholine receptors. So, nicotinic acetylcholine receptors are present in the subneural cleft. Next, we'll move on to neuromuscular transmission. What is neuromuscular transmission? So, neuromuscular transmission is defined as the transfer of information from motor nerve ending to the muscle fiber through the neuromuscular junction. So, it is mechanism by which motor nerve impulse initiate the muscle contraction. So, what is the function of neuromuscular transmission? When the information is being transformed from a motor nerve to a muscle fiber to cause or to initiate the contraction of the muscle is being brought by a neuromuscular transmission. How this neuromuscular transmissions are brought? These are brought by some 5 events like namely release of acetylcholine, action of acetylcholine, development of end plate potential, next the development of miniature of end plate potential, in conclusion that is destruction of acetylcholine. First, we'll see the release of acetylcholine. When the action potential reaches the axon terminal. What is action potential first of all? Action potential is nothing but an electrical signal that propagates along the surface of the membrane of neuron due to the movement of ions. Ions like sodium and potassium between the interstitial fluid and inside of a neuron through a specific ion channel in the plasma membrane. In simple, action potential is nothing but due to the change in the membrane potential, there is a creation of action potential. So, when action potential reaches the axon terminal, it causes the opening of an voltage gated calcium channel in the membrane of axon terminal. Calcium ion from extracellular fluid, that is ECF, enters the axon terminal. So, when calcium ion uptake into terminal causes release of neurotransmitter acetylcholine into synaptic cleft, which has been synthesized, stored into the synaptic vesicle. So, these causes bursting of the vesicles by forcing the synaptic vesicles move and fuse with the presynaptic membrane. Now, acetylcholine has been reached the synaptic cleft and released from the ruptured vesicle. By exocytosis, acetylcholine diffuses through the presynaptic membrane and enters the synaptic cleft. So, acetylcholine travels across the synaptic cleft to the postsynaptic membrane. That is, which is also known as motor end plate. In simple, in release of acetylcholine, when action potential reaches the terminal, it causes opening of voltage gated calcium channel and release of acetylcholine into the synaptic cleft. The second event is action of acetylcholine. After entering the synaptic cleft, acetylcholine molecule binds with a receptor, namely nicotinic receptor, in the postsynaptic membrane and form a complex, namely acetylcholine receptor complex. So, it increases the permeability of postsynaptic membrane for sodium by opening the ligand-gated sodium channel. So, as we could see in this diagram, acetylcholine molecule combine with the receptor, that is nicotinic receptor, and form a complex, namely acetylcholine receptor complex, and cause ligand-gated sodium channels to open. So, which causes influx of sodium from ECF to the interstitium. Now, sodium ion from ECF enters to the neuromuscular junction through these channels, and the sodium ion alters the resting membrane potential and develop an electric potential called end plate potential. The next event is the development of end plate potential. So when there is a alter in the resting membrane potential, there is a development of electric potential which is known as end plate potential. So when there is a change in resting membrane potential, when an impulse reaches the neuromuscular junction. Okay, so the resting membrane potential at neuromuscular junction is minus 90 MV. But when the sodium ion enters inside, that is from ECF to the interstitium, this causes depolarization. Depolarization means a slight change in the polarization occurs up to minus 60 MV, which is called end plate potential. So in simple, end plate potential are voltage which causes depolarization of skeletal muscle fiber. It's caused by neurotransmitter binding to the postsynaptic membrane. That is, when the acetylcholine binds to the postsynaptic membrane in the neuromuscular junction, there is a slight depolarization, which is known as end plate potential. And this end plate potential is non-propagative. That is, it does not spread and it causes the development of action potential in the muscle fiber. Next event is the development of miniature end plate potential. It is the weak end plate potential in the neuromuscular junction that is developed by the release of small quantity of acetylcholine from axon terminal. And each quantum of this neurotransmitter produces a weak miniature end plate potential. The amplitude of this is 0.5 MV. So, this miniature end plate potential cannot produce action potential because of its low quantum. But when more and more quanta of acetylcholine are released continuously, this miniature end plates are added together and finally it produces an end plate potential. In simple, miniature end plate potential does not causes acetylcholine, that is release of acetylcholine, but when there are more quanta of acetylcholines are released continuously, this miniature end plate potential develops into an end plate potential, resulting in action potential of the muscle. So, till now, what we saw, the events in neuromuscular transmission, we'll just revise. First, in motor nerve fiber, action potential is being generated, and the next event takes place in axon terminal, that is, opening of voltage-gated calcium channel and entry of calcium ion from ECF causes opening of synaptic vesicle and release of acetylcholine. So now the acetylcholine moves into the synaptic cleft. From the synaptic cleft, it binds of acetylcholine with the receptor known as nicotinic acetylcholine receptor in the postsynaptic membrane, causing formation of acetylcholine receptor complex. Now, it causes opening of ligand-gated sodium channel and entry of sodium ion from ECF, development of end plate potential. Next, the events takes place in muscle fiber like generation of action potential, excitation and contraction coupling, and in final way, the muscle is being contracted. So, the last event in the neuromuscular transmission is destruction of acetylcholine. Acetylcholine is released into the synaptic cleft is destroyed very quickly, that is within 1 millisecond by the enzyme acetylcholine esterase. However, acetylcholine is so potent that even in the short duration of 1 millisecond is sufficient to excite the muscle fiber. So, which means a rapid destruction of acetylcholine is needed so that it doesn't cause a repeated excitation of muscle fiber. So, it prevents repeated excitation of the muscle fiber and allows the muscle to relax. So that is the main function of rapid and quick destruction of acetylcholine to prevent the repeated excitation of the muscle fiber. So with this we have concluded the neuromuscular transmission and its events. Next we will move on to neuromuscular blockers. What is neuromuscular blockers? Here comes the pharmacological effect which acts in the neuromuscular junction. These are the drugs which prevents the transmission of impulse from nerve fiber to the muscle fiber through a neuromuscular junction. So we'll see some kind of drugs like Cure. Cure prevents the neuromuscular transmission by combining with the acetylcholine receptor. So these blockers are known as receptor blockers. Next is bungarotoxin. It is also a neuromuscular transmission blockers by blocking the acetylcholine receptor. Next drug is succinylcholine or carbamylcholine. This is a drug which blocks the neuromuscular transmission by acting like acetylcholine and keeping the muscle in depolarized state. But these drugs are not destroyed by cholinesterase, so the muscle remains in depolarized state for a long time. Next is botulinum toxin. See, this is derived from a bacteria Clostridium botulinum, and it prevents the release of acetylcholine from axon terminal into the neuromuscular junction. Next and last, we'll see about applied physiology. Applied physiology is nothing but the disorders of neuromuscular junction. The two main disorders we should remember is myasthenia gravis and Eaton-Lambert syndrome. Myasthenia gravis is an autoimmune disorder caused by antibodies to cholinergic receptor. That is, antibodies are produced in our body to destruct or destroy the acetylcholine receptors in our body. Next is Eaton-Lambert syndrome. It is also an autoimmune disorder of neuromuscular junction caused by production of antibodies to the calcium channels in axon terminal. So, our own body produces the antibody against the calcium channel which is being present in axon terminal. So, we have come to an end. We'll just recall what we saw in this video. First, what is neuromuscular junction? Neuromuscular junction is a junction between the muscle fiber and the motor neuron. And we saw anatomical structure like axon terminal, that is also known as motor end plate, synaptic trough or gutter, synaptic cleft, subneural cleft. Next, we saw about neuromuscular transmission, which is most important, that is defined as the transfer of information from motor nerve ending to the muscle fiber through neuromuscular junction. It is a mechanism by which motor nerve impulse initiate the muscle contraction, and events in this neuromuscular transmission like release and action of acetylcholine, development of end plate potential, development of miniature end plate potential, destruction of acetylcholine. In last, we saw some neuromuscular blockers and some conditions like myasthenia gravis and Eaton-Lambert syndrome. I hope you all enjoyed this video and understood about what is neuromuscular junction. Thank you.